DNA elongation is the stage of DNA replication where DNA polymerase adds nucleotides to the 3' end of a new strand. This step is essential for cell division and ensures that each daughter cell receives an identical copy of the genetic information. As discussed in the previous video, link in the description, replication begins with the initiation stage, which occurs before elongation. During initiation, helicase opens the double helix at a specific site called the origin of replication, unwinding the parental strands and forming a replication bubble with 2 forks at either end. Single-stranded DNA binding proteins, such as replication protein A in eukaryotes, attach to the separated strands to prevent them from re-annealing. Next, primase adds short RNA primers, providing DNA polymerase with a starting point, since the enzyme can only add nucleotides to an existing 3' end and cannot start synthesis on its own. As helicase continues to unwind the helix, the DNA ahead of the fork becomes positively supercoiled or overwound. If this strain isn't relieved, the replication fork would stall and the DNA could even break. To prevent this, cells use topoisomerases, enzymes that relieve torsional strain by cutting and rejoining the DNA strands. In eukaryotes, topoisomerases 1 and 2 perform this role, while in bacteria, DNA gyrase, a type of topoisomerase 2, carries out the same function. These enzymes temporarily break one or both strands, allow controlled rotation or strand passage, and then reseal the DNA, enabling helicase to continue unwinding smoothly. Before elongation can proceed efficiently, DNA polymerase must remain firmly attached to the DNA template. This stability is ensured by sliding clamp proteins, which form a ring around the DNA and hold the polymerase in place, allowing it to add thousands of nucleotides without falling off. In eukaryotes, this clamp is called pCNA, proliferating cell nuclear antigen, and in bacteria, it's known as the beta clamp. These clamps are loaded onto DNA by clamp loader complexes, replication factor C in eukaryotes, and the gamma complex in bacteria. These complexes use ATP to open the clamp, place it around the DNA, and release it to engage the polymerase. Once assembled, DNA synthesis proceeds bidirectionally, with each helicase moving towards the opposite fork from the origin. DNA polymerases extend the primers, adding complementary nucleotides one by one. After each addition, the enzyme shifts forward by one base pair, a process known as translocation, positioning the active site for the next nucleotide. In bacteria, DNA polymerase III performs this function, while in eukaryotes, the main enzymes are DNA polymerase epsilon and DNA polymerase delta. All three possess 3' to 5' exonuclease activity, allowing them to proofread newly added bases and maintain replication accuracy. This means the enzyme not only has a polymerase active site that adds nucleotides, but also an exonuclease site that detects mismatched bases at the 3' end, removes the incorrect nucleotide, and allows the polymerase active site to replace the incorrect nucleotide with a correct nucleotide. Because DNA polymerase can only build new strands in the 5' to 3' direction, the two template strands are copied in opposite ways. The template strand running 3' to 5' towards the replication fork can be copied smoothly. DNA polymerase moves along it continuously to form the leading strand. The opposite template strand, which runs 5' to 3' towards the fork and therefore 3' to 5' away from it, must be synthesized in short separate fragments. These short pieces, called Okazaki fragments, are later joined together to form the lagging strand. The leading strand requires only a single primer, while the lagging strand needs multiple primers, since new fragments must begin each time the helicase exposes more template DNA. In eukaryotes, a polymerase alpha primase complex synthesizes short RNA DNA primers, which DNA polymerase delta then extends, while DNA polymerase epsilon synthesizes the leading strand. In bacteria, primase DNAG makes the primers, and DNA polymerase III extends both strands. Once the Okazaki fragments are completed, the RNA primers must be removed and replaced with DNA. In eukaryotes, DNA polymerase delta extends the upstream fragment, displacing the RNA DNA primer into a short flap. This flap is cut off by flap endonuclease 1, FEN1, then DNA ligase 1 seals the remaining nick to create a continuous strand. In bacteria, DNA polymerase 1 removes the RNA primers and fills the gaps, and DNA ligase joins the fragments together. After this processing, each newly replicated region becomes continuous, though overall replication continues until the termination stage, which will be discussed in the next video. Once replication is underway, in eukaryotic cells, another important aspect comes into play: the handling of chromatin. Because DNA is wrapped around histone proteins to form nucleosomes, these structures must be disassembled ahead of the fork and reassembled behind it. Parental histones are redistributed between the daughter strands, while new histones are added and modified to match the parental pattern. Enzymes copy these histone marks, and maintenance methyltransferase restores DNA methylation, ensuring that chromatin structure and epigenetic information are faithfully preserved. This allows cells to maintain stable gene expression patterns after replication. So in this video, we explored the elongation stage of DNA replication, how DNA polymerase extends new strands by adding nucleotides to the 3' end. We saw how helicase, primase, sliding clamps, and topoisomerases work together to ensure smooth and accurate replication, how the leading and lagging strands are synthesized, and how Okazaki fragments are processed. Finally, we saw how eukaryotic cells manage chromatin and DNA methylation during replication, ensuring that both the genetic sequence and the epigenetic information that regulates it are accurately passed on.